Okay, hello everyone. Good afternoon, all of you. Good afternoon. Okay, so let's continue. In the last class, we were discussing about if everyone quickly recall, we were discussing about the microbial physiology, and in the last class, friends, we started discussing about the growth curve. Is it? Okay, good afternoon. So when we were discussing about the growth curve, we understand about different type of a growth curves, that is the lag phase, log phase, as well as the stationary phase, as well as the decline or death phase. Till there we all people has studied. Okay, now all of you just listen to me carefully. Now what we do is, friends, we will do we will finish the left part of the microbial physiology, and just listen to me carefully. In the cell biology chapter, I told to all of you, friends, I will teach only 4 topics. That is, see everyone listen to me. So I am teaching, so far what we have discussed is, we have discussed the transposons, we discussed, and we have discussed, friends, cell cycle and its regulation, and we have also discussed about the microbial physiology, and we are going to finish it. Okay, now the 4th topic, the most important topic, which was repeatedly asked in the exam in terms of 4 marker, is the ER. Okay, especially the RER. In the RER, especially what the question they have asked is, good afternoon everyone, what the question they have asked is protein sorting. Okay, protein sorting and the process of glycosylation. Friends, these are all the topics that they have asked very, very oftenly. Okay, good afternoon everyone, good afternoon everyone. So please understand. So in the today class, good afternoon everyone. In the today's class, we will finish the microbial physiology, and after finishing the microbial physiology, friends, we will go and discuss about the even ER also. Okay, so now let's try to understand one by one, briefly and elaboratively. Okay, now let's start it. Okay, good afternoon everyone. Let's continue. Okay, so in the last class, friends, if you quickly recall, we have understand about we just started microbial physiology, and we have understand about the 4 major phases of the growth curve. And friends, I hope every student clearly remember that what is exactly the dioxic growth curve. I have very clearly told to all of you, friends, dioxic growth curve is possible only whenever there are 2 sugars, or whenever the 2 sources are available. And I have clearly told to all of you, friends, when the 2 sources are available, please kindly don't think that the bacteria using the 2 sources at a time. It never happens. It never use the 2 sources simultaneously, friends. Even we have discussed in the question also. What we discussed, friends, it will use the 2 sources sequentially only. First it use one source, then followed by the another source. I hope everyone remember that. And friends, I requested all of you to remember the most important events that are happening in the stationary phase. If everyone quickly recall, my friends, in the starvation phase, what did I tell you, friends? The bacteria is going to synthesize the starvation proteins. Good afternoon everyone. And the bacteria is synthesizing the DPS. Bacteria is increasing their peptidoglycans linkase, and most importantly, bacteria is synthesizing the chaperones also. And friends, what we discussed, because of all these, what is going to happen, friends? Because of all these, the bacteria will become resistant in such a way that, friends, bacteria can survive up to few years in the phase of starvation only. And friends, when we are discussing about the death phase, what we understand in the death phase, I have clearly told you that, friends, death phase is also what we discussed. We understand that the death phase is also logarithmic only. Why I have used, friends, death phase is also logarithmic? Because we understand that all the bacteria are not dying at a time. Please don't get confused with this particular point. What is the statement I have used to you? That at a particular time, everyone remember to me, at particular time only the particular number of cells are getting died. So that's why friends, we will be using the word that death also logarithmic fashion only. And I have also highlighted that friends, how do we come to know whether the bacteria died or not? What is the only method? So the major method that is used to define whether the bacteria died or not, what we will do friends, we will take that particular bacteria and we will keep it in the fresh medium. Correct na friends? We'll take the bacteria and we will keep it in the fresh medium. Whenever we keep the bacteria in the fresh medium, every student remember, an example, if it is not happening, listen my statements very carefully, if it is not dividing, if it is not preparing, then we assume that the bacteria is died. So in single word we can define the death as friends, irreversible loss of the ability to reproduce. That is what we can call friends death. And if you very quickly recall friends, we have solved this particular question and we have also solved this particular question also, that is a dioxic pattern of biomass. Okay, so now friends, first I will explain the remaining concept, then we will go to the ER and then we will solve the numericals. Okay, now every student please listen to me very carefully. Friends, this slide is a very important slide. Okay, 2 times we got the question from this particular slide specifically friends, and both time we got the question in the part C only, that is for the 4 marker. I know you can't see this particular slide clearly, but friends, in the app once you see the PPT you can see it very clearly. And in this particular slide it is, there is no concept to explain to all of you, but I don't know friends, I want you people to remember, good afternoon, I want you people to remember whatever I am exclusively marking. Once again I am requesting you friends, please don't neglect this kind of questions. They are very easy questions, but the people cannot answer. The simple reason behind that, they never ever study this. They always they tend to skip this or they tend to avoid this. But I am requesting all of you to read this without fail. Here you have to use only your memorizing skills. Please try to remember as many as examples, especially remember whichever I am marking. Now friends, this microbial responses or this particular classification was actually based on different type of terms. Based on what friends? Based on their activity. What do you mean by that? So how the bacteria are surviving at a very high saline condition, and how the bacteria are surviving at different pHs, how the bacteria surviving at different temperatures, as well as how does the bacteria surviving at different oxygen concentration as well as the pressure. I am requesting you friends, whichever I am marking, please remember without fail. So now listen to me carefully. I will ask you in the tomorrow class also. So the first one we can call the osmotolerant. What do you understand by the word osmotolerant? Everyone remember, osmotolerance means friends, able to grow over wide range of water activity. Remember friends, osmotolerant means it doesn't matter whatever the water condition it may be. Even though water condition is the higher, that particular bacteria having the ability or capability to survive at that higher water conditions. That's what we can call it as the osmotolerant. And I want you to remember this particular example without fail friends, that is the Staphylococcus aureus. Please remember only this one example, more than sufficient. Now halophile. Halo means what friends? The organisms which require the sodium chloride, high salinity condition. And friends, please kindly don't think that the bacteria can grow at any kind of a salinity condition. That is also not correct statement. Every bacteria having particular salinity conditions only. Some bacteria above that level can grow, some bacteria above that level cannot grow, some bacteria below that level cannot grow, some bacteria below that level they may grow. So it will be varied from one bacteria to the another bacteria. But generally friends, I want you to remember only this one example friends, you don't need to remember extra. The major one, halobacterium. Please remember friends, what is that bacterium? Halobacterium. So this, if this particular bacteria wants to grow, every time friends, the condition should be always 0.2 molar to grow. Please kindly do not get confused with this particular statement. And friends, now please listen. You have to answer one question right away. Please focus. pH. We all know friends, we use it to define bacteria are ubiquitous in nature. Friends, what do you mean by the word ubiquitous in nature? I have told to all of you many times friends, some bacteria can grow only at acidic temperature, some bacteria can grow at basic temperature, some bacteria can grow especially at the neutral temperature or the neutral pH. Especially the pH. We cannot say that every bacteria can live only in the acidic pH. Depending on the environmental condition, bacteria changing their pH levels also. So let's understand, if a particular bacteria, if it is growing in between the pH 0 and 5.5, then friends, we will be calling it as the acidophile. If a bacteria is growing at the pH 5.5 and 8, we will be calling it as the neutrophile. In simple word, whichever is living under the neutral pH. Please be patient friends, lot of things to be remembered. And whatever the bacteria that is living at the basic pH, that is 8.11 and 8.5 and 11.5, this is called as alkalophile. Friends, please listen. Here I want you people to remember many examples. The acidophile, I am marking friends, whichever I am marking, please remember without fail. Sulfolobus, ferroplasma, aconitum. I want you to remember these three examples without fail. Sulfolobus, ferroplasma and aconitum. Please don't confuse. Then friends, neutrophiles, which are living in the neutral pH. Please remember three examples. I want you to remember three compulsory. Escherichia coli, Euglena and Paramecium. Whereas alkalophile, which is growing in between 8.5 and 11.5, remember only one example, only one. That is the Bacillus alkalophilus. Easy to remember also friends. Alkalophile, and the example is Bacillus alkalophilus. Very easy to remember. Now friends, temperature, every student remember, under the temperature there are many are there. This is the one which was asked in our previous year exam. Psychrophile, psychrotrope, mesophile, thermophile, hyperthermophile. Sometimes friends, they won't give the example also. In the exam, they won't give the example. Instead of giving the example friends, they will directly ask what that particular organism meant to be. What do you mean by the word mesophile? What do you mean by the word the thermophile? And at which particular temperature they are surviving? That also we can expect in the exam. So friends, there is a minute differences between all. Focus everyone. When you use a word psychrophile, what's the common point has to come in your mind friends? Please remember, it can grow abundantly at 0 degree centigrade. It can grow abundantly at 0 degree centigrade, but friends, it can grow up to 15 degrees centigrade, or I can use the word less than 15 degrees centigrade. What do you mean by that? If you keep a psychrophile at the temperature above 15 degrees centigrade, friends, it cannot grow or it cannot develop. Always remember that. So psychrophile, in order to grow, it requires 0 degrees minimum. It can grow abundantly at the 0 degree, but up to 15 degrees it can grow very easily, but above 15 degrees friends, it cannot grow. Now when you come to the psychrotrope, every student remember, it can tolerate 0 to 7 degrees centigrade. Please kindly don't get confused with that point. And when I use the word optimum, you have to remember optimum 20 to 30 degrees centigrade. Okay, now next one. Friends, please don't get confused between the mesophile, thermophile and hyperthermophile. I want you to listen to me very carefully. Friends, the moment you heard the name of a mesophile, please remember friends, mesophile are the type of bacterium, they can also adjust like humans. What I mean to say, let's consider ourselves only. We can't survive for the longer time if the temperature is below 20 degrees centigrade. We can't adjust for very long. I am using the word very long time. And at the same time 45 degrees centigrade, if the temperature is repeatedly 45 degrees centigrade, our survival ability will be automatically decreases. Same, mesophiles are also same, friends. They are having the ability or capability to grow optimum between these temperature. So if it is no, it is not past. Okay, if it is below 20 degrees centigrade, it cannot grow. If it is above 45 degrees centigrade, it cannot grow. And friends, thermophile, all the students keep it in your mind, thermophile can grow at 55 degrees centigrade or higher. See friends, there is a difference between the thermophile and hyperthermophile. Please don't get confused. Thermophiles, friends, they can't grow above 65 degrees centigrade. They can't grow above 65 degrees centigrade. But friends, if you look into the hyperthermophile, it can grow up to 113 degrees centigrade also. Friends, the hyperthermophiles can grow very easily. So remember, mesophiles are up to 20 to 45. Thermophiles are 55 and 65. Hyperthermophiles are up to 113 degrees centigrade. Please don't get confused. And friends, you have to remember the temperatures also without fail. And most importantly for the exam is examples. Friends, whichever I am marking, please remember. Okay, so psychrophile, which is living at the 0 degree centigrade. Bacillus psychrophilus, please remember that. Psychrotrope, which is growing at 0 to 7 degrees centigrade. Please remember Pseudomonas fluorescens. Whichever I am marking, I want you to remember only that. Mesophiles. Please, for the mesophiles, friends, I want you to remember the 3 examples without fail. Please be patient. E. coli, Neisseria gonorrhea, Trichomonas vaginalis. The Trichomonas vaginalis, which can cause the fungal infections. Neisseria gonorrhea, which can cause the gonorrhea. E. coli is the generalized bacteria. These 3, please remember, they are all the mesophiles. And friends, thermophiles, we all people use it to study in the PCR technique. So please remember the example, Thermus aquaticus. One is enough. Remember Thermus aquaticus. And friends, for the hyperthermophile, I want you to remember one example, that is Sulfolobus. Please don't forget. Sulfolobus is the type of bacteria which can grow up to 130 degrees centigrade. Friends, you know, if you keep the Sulfolobus at lower temperatures in autoclave also, if you keep the Sulfolobus at lower temperature in hot air oven also, it can survive up to particular extent, right? So it can easily tolerate up to 113 degree centigrade. So now we have divided the bacteria, friends, based on solute and water activity, based on pH, based on temperature. Now friends, we are concentrating on oxygen concentration. This is also extremely important. I want you to pay attention. Everyone remember. First, remember the definitions, friends. Remembering the term definition is important. Obligate aerobe. Friends, what do you mean by the word obligate? We all people know very well that obligate means compulsory. Obligate means necessary. Obligate means compulsory or necessary. So when I use a word obligate aerobe, what you have to understand, that particular bacteria compulsory, mandatorily, it requires the oxygen. Without oxygen, that bacteria cannot grow or cannot survive. So if they want to grow, there will be a requirement of the oxygen. And I want you people to remember 3 examples: Pseudomonas, Mycobacterium, fungi and protozoa. Remember 4. Pseudomonas, Mycobacterium, fungi and protozoa. Don't forget. Next one, facultative anaerobe. Friends, I have told you many times about the word facultative. Facultative means, here I have used a word facultative anaerobe. What do you mean by that? Whether, every student remember my words, whether the oxygen is present or absent doesn't matter, friends. Whenever there is a presence of oxygen, it can grow abundantly. But whenever there is a no oxygen, in the absence of oxygen, it will grow, but the growth is not abundant, friends. What I can use the word, in the absence of oxygen, they will just grow. But the point is here, in the presence of oxygen, friends, they will grow much better. Please do not get confused with this particular point. Next one, aerotolerant anaerobe. See, I am using a word, friends, aerotolerant. What do you mean by the word tolerant here? I am using the word tolerant because the growth is equal. Friends, there is a great difference between the facultative and aerotolerant. Most of the students are getting confused between that. Facultative means in the absence of oxygen it will grow, but in the presence of oxygen the growth is abundant, extremely higher growth. But when I use a word aerotolerant anaerobe, every student remember, it will grow equally in the presence of oxygen also, in the absence of oxygen also. The best example, Streptococcus pyogenes, I want you to remember. Now, in the first, I told you that, friends, obligate aerobe, which compulsory require the oxygen for its growth and development. Now, I am using the word, friends, obligate anaerobe. What is obligate? Compulsory. What is anaerobe? Do not require the oxygen. What do you mean by that? Obligate anaerobe means, friends, if example, if there is a oxygen, it will die immediately. Which means, friends, obligate anaerobe means does not tolerate the oxygen and it will die in the presence of oxygen. Best example, friends, methanobacterium and clostridium. These two are the most important bacteria. Next one, microaerophile. Micro, very low concentration. Aero, oxygen. See, I am not saying that, friends, they do not require the oxygen. They require the oxygen, but the condition is, friends, they require a very less amount of the oxygen. If oxygen is more, once again they will die. See the condition. If the oxygen level is higher, they will die. But in the absence of oxygen also they will die. Then how they will grow? Friends, they will grow only when the oxygen level is extremely lower. Low the oxygen level, they will survive. So it requires oxygen level below 2 to 10% for growth. So if the oxygen level is more than 10%, it immediately die. Best example, please remember, friends, Trypanoma palladium. I don't know what you do. You can take the printout, you can, whatever you do, you can make your own notes also. In just 15 minutes you can make it off. I don't know what you do, and I want you to remember it compulsory because it is extremely important for the exam. Okay. Now everyone answer this question. Don't be hurry, just think and answer the question which was asked in the February 2022 in the part C. Okay, even though you know the answer, doesn't matter for me. I want you to pay attention. Even though you know the answer, friends, I want you to think it. Even though you know the answer. And I want everyone to give me answer, please. I want everyone. I am requesting everyone to give the answer, everyone. Everyone. Apurva said C. Anyone else? Others, you know, okay. That's great. Okay, see, I never say that luck will work all the time, but in this kind of questions the luck will work. At least if you know one single word, that is what is a hyperthermophile. If you at least you know this hyperthermophile, 4 marks in our hand. We all know that, friends, hyperthermophiles are the one which are extremely living at extremely high temperature. So obviously you can easily come to a conclusion for this particular option C. Very easy it is. Right. And friends, what is psychrophile? We discussed now, which is living at the 0 degree centigrade, extremely cold temperature. Hyperthermophile at extremely high temperature, and we also know very well alkaliophile, high alkaline environment. And the only thing which you don't know is the hyperpiezophile. Friends, sometimes instead of giving the hyperpiezophile, they will directly use the word barophile. Okay, they will use the word barophile. Anything is same, friends. Barophiles are also a type of bacteria which are basically surviving at the higher pressure. Barophile is same or hyperpiezophile is same. Please don't get confused with that. And we already studied the first one, halosaline, which is living at the high salinity condition. Hence, friends, the correct answer will be obviously C. See, such a simple 4-marker question which we got in the 2022. Okay, anyway, tomorrow I am conducting one session. Okay, so tomorrow is what date, friends? Tomorrow is 16th, right? So tomorrow is the 16th of August, and our exam will be in between 16th to 19th September. So what I do is I will just give 1 month timetable. Okay, 1 month timetable I will give for all of you, and I am not at all saying to follow this particular timetable only strictly, but I can give the most feasible and possible timetable. I will give 2 different type of timetables, so whichever is easy and convenient to all of you, you follow that. But I am just requesting is try to follow at least one method. And the most important word, this timetable will be applicable only for the students who already prepared. Whomever preparing very newly, it's a kind of difficulty for them, but I want you to give the try. Okay, anyway, that we will see in the tomorrow class. Okay, now let's continue. Okay, so friends, we understand about the different type of bacteria, right? Now friends, let me quickly explain you little bit about the bacterial growth pattern. Okay, here sometimes we can expect the part B questions from the name of the proteins. Okay, from the name of the proteins we can get the sometimes part B question. Okay, I'll quickly finish this. Okay, everyone just pay attention. Bacterial growth. I have told to you, friends, bacteria is a single-celled organism. What is the statement I have given to all of you? Whenever a microbiologist wants to understand the growth of a microorganism, it is really difficult for us to study each and every single bacteria. That's the reason what we will do, friends, instead of reading one single bacteria, we will always read the group of microorganisms. Once we study the group, friends, it is very easy to understand their particular growth. Friends, I was continuously repeating the word bacterial growth. But the question is here, friends, how can I define the word growth? In one simple word, the growth equates to the cell reproduction. Correct, na? See, example, everyone please imagine we are a multicellular organism. In the multicellular organism, if the growth is happening or not, if I ask you the growth is happening or not, how can you tell me? You can easily tell me that the growth is happening exclusively based on the size of the organism. If the size of the organism is increasing, then we can conclude that, of course, friends, yes, there will be a growth in the multicellular organism. But When you look into the prokaryotic organism or the unicellular organism, especially friends, there is only one cell. Then one cell having a particular size only, then how can we say whether the growth is happening or not? In this particular condition, friends, the growth can be determined by the number. So increase in the number of individuals in the population, that is what we will consider in case of a unicellular, and here we will consider with respect to the multicellular, we will consider with the size in the multicellular, number in the unicellular. I will quickly summarize this in 5 to 10 minutes, and I will tell you what kind of protein names you people has to remember for the exam. We have studied from the 11th and 12th standard only, friends. There are different kinds of reproductive methods followed by the bacteria, but we commonly know that bacteria can majorly follow the asexual reproduction processes. It can follow the binary fission, it can follow the multiple fission, it can follow the endospore formation method, as well as it can also follow the budding method. But friends, among the binary fission, multiple fission, endospore formation, as well as the budding, what bacteria majorly prefer, friends? Most of the time, friends, bacteria will prefer only the binary fission. What is the reason? What is the reason the bacteria only preferring binary fission when compared to all other methods? The simple reason behind this is binary fission is a very simple method where just one cell or a single cell getting split into two equally sized cells, and it's a very easy process. Most important reason, friends, there is an involvement of less proteins. Less proteins or the less enzymes. That's why when compared to the budding and spore formation and multiple fission, bacteria always follow the binary fission only. Now I am going to tell you, friends, what kind of proteins that are basically involved in this. That only you have to remember for the part B. Focus. Okay, so friends, you people understand what's happening in the binary fission. The first step, every student remember, first step, cell wall get elongated. Very simple point. And the second step, the DNA get separated. And the third step, the cytoplasmic content also separated. Then obviously, friends, the one parent cell is getting split into two equal daughter cell. Now I'm going to explain you what kind of proteins are required for the cell wall elongation. How does the cytoplasmic elongation or splitting is taking place? Just listen to me, the overview. Understand? Friends, we all people clearly studied when compared to the cell wall and cell membrane. What did I tell you, friends? Which is the most living part? Please don't get confused with my words. I am using a word most living part. When I use a word most living part, which means most active part metabolytically. So which part of the bacteria will be more active in terms of metabolism? Undoubtedly, friends, cell wall. Most of the students will think that the plasma membrane is performing the many functions, so that's why it is more important. No. First of all, friends, if the plasma membrane of the bacteria wants to perform many function, the plasma membrane has to be protected compulsory. How does it get protected, friends? Of course, with the help of a cell wall. So what I want to conclude here, friends, in the binary fission, listen everyone, in the binary fission, the binary fission starts with elongation of the cell wall. Every student remember, first the cell wall get elongated. Friends, keep it in your mind, whenever the cell wall is getting elongated, friends, undoubtedly the cell membrane also get elongated, because cell wall is the most rigid structure. If cell wall elongated, membrane automatically elongated. Whenever the membrane is getting elongated, every student please remember, the DNA get duplicated, friends, and the duplicated DNA starts moving. Okay, it is moving towards what? Friends, it is important. It is moving towards the edges. It is moving towards the edges. And every one of you keep it in your mind, friends, one cell wants to split into two cells. It is extremely important, friends, there should be a formation of a septum. without septum formation, one cell, one parent cell never split into two. Now you can ask me, sir, what exactly the septum is doing? So friends, the septum will be, every student remember, the septum will be helpful in the membrane invagination. What friends? The septum is actually involved in what process? Membrane invagination. You all people can see here, friends. See here, this is the septum. The septum will go deeper, 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 and it can split one cell into two cells. Now, how does this particular septum will be formed? What kind of proteins that are involved in the septum formation? We will study little more elaboratively. Okay, I just given you the overview of the binary fission, and majorly we will study what kind of proteins involved, because these are all important for part B. Now let me give you the overview of the other processes also. Now, what is multiple mitosis? You all people can see here, friends. The cytoplasmic content is same. Friends, remember, the cytoplasmic content is same, which is getting divided continuously, friends. Of course, you can see here the nucleus is getting divided. So, and every nucleus can be surrounded by a cytoplasmic content. In a simple word, first nuclear division occur, which we can call it as karyokinesis. First, there is an occurrence of the karyokinesis. Immediately, friends, there is an occurrence of the cytokinesis. And the point is here, friends, this multiple mitosis or nothing but multiple fission, this process is a time-taking process. That's why bacteria never prefer this kind of processes. And this is the most important process, friends. When the bacteria is going to prefer the this endospore formation method, everyone has to remember, friends, if a bacteria is in the adverse conditions, every student remember, if the bacteria is in the adverse conditions, everyone remember, then only they can form the endospore. An example, let's take an example of drying. Let's take an example of drying or the desiccation. Friends, what do you mean by the word desiccation? I told to all of you, desiccation is the condition where there is absolutely no water content. Inside the bacterial body, the water content is zero. Literally zero, the water content. So friends, there is a chance that the bacteria may die. Only during that unfavorable condition, the endospore formation occur. Otherwise, friends, the endospore formation never takes place. Okay, every student, please listen my words very, very carefully. See, once the condition is unfavorable, what's happening, friends? Immediately you can look into this. This is the major DNA of the bacteria, and this is the extracircular DNA. We all people know that, plasmid. Now what is happening, friends, in the unfavorable condition? The original DNA is getting duplicated, friends, as well as plasmid is also getting duplicated. There is no doubt about this. Now friends, I want you people to remember the word very carefully. Once the duplication is over, everyone pay attention, friends. The endospore is getting formed. Please remember the statement. Endospore is getting formed, friends, only around the replicated DNA. Please remember this statement. Endospore is getting formed only around the replicated DNA molecule. Now what is happening, friends? Now an example, the condition is desiccation. What happens? Bacteria can't survive for the longer time. Now you just imagine, friends, the bacteria died. Simple point. Now bacteria died. When the bacteria died, what happened, friends? From that particular died bacteria, what is getting released, friends? Endospore is getting released. Now you can already see that endospore is already covering the original DNA and extracircular DNA, that is the plasmid. Now what is happening? Once the conditions become favorable, immediately what is happening? Immediately, friends, this particular DNA and plasmid, which leads to the formation of a new bacteria. So this is how only during the adverse or the unfavorable conditions only, friends, the endospore is getting formed. Please kindly do not get confused with that particular point. Now budding is a very simple process, won't occur every time. See, most of the time bacteria only follow the binary fission Okay, and in adverse conditions, what we can use the word endospore. Now, whether it is the budding or the multiple fission, friends, always keep it in your mind. They are not specialized processes. They are very generalized process. But most common process that always occur is binary fission only. And we all people know that, friends. We read in the 11th standard only. In the budding, what happens? There will be a bud starts begin on the top of the parent cell. Now you all people can see, right, there is a nucleus is present here. Now friends, the nucleus undergo the duplication, nothing but nucleus get copied. Friends, whatever the nucleus that get copied, see here, now the bud receive the copy of the nucleus. Friends, the bud not only receiving the copy of the nucleus, friends, the bud is also receiving the cytoplasmic content. Now you only understand, it received the nucleus which contain the most of the information, and cytoplasm also we know it also contain abundant amount of enzymes. So now it received the nucleus and received the cytoplasm. Now it is ready to develop as an individual cell. So now it is separated as the daughter cell. Very simple point it is. Like that, now you remember, now it become parent cell. Now it also form another bud. Like that, friends, there are chain of cells, or like that there are group of cells are getting formed. Please kindly do not get confused with that particular point. Okay, now we have to read majorly about the binary fission and proteins involved in the binary fission. Proteins is important. Okay, focus. Everyone pay attention. See, all the students, please look into the diagram very carefully. Here you can see, friends, very clearly. The outer yellow color layer is the cell wall, and the inner light pinkish color is the cell membrane. And you can see this is the chromosome, and these are all dotted like structures. You can see, friends, they are the ribosome. Now you try to understand, friends, entire cell never elongated immediately. All the students keep it in your mind. First, there is a elongation of the cell wall. See, there will be a no requirement of a specialized protein. Every student remember, there will be a no requirement of a specialized protein for the cell membrane elongation. When the cell wall get extended, friends, automatically cell membrane get extended. Remember that point. See here, cell wall extended and cell membrane is also getting extended. Now please listen my statement. Friends, the moment cell wall and cell membrane got extended, everyone remember, friends, there is a formation of the septum. Okay, now what is the statement that I want you to remember for the exam? Listen to the statements carefully. The septum formation is giving an indication that the chromosomes are moving towards the opposite ends of the pole. Once again, listen my statements very carefully. I am saying, friends, septum formation, everyone remember, septum formation bringing the two chromosomes, bringing the duplicated chromosomes towards the end of the cell. Now please remember, whenever the chromosomes are moving, friends, apart from the chromosome, I want you people to remember that ribosomes also moving, as well as, friends, cytoplasmic content is also moving. Now all the students remember, once again I am repeating my statement. My first statement is septum formation is a key event that is indicating the chromosomes are moving end of the poles. First statement, septum formation. Now you know what is the second statement I am giving to all of you, friends? Septum completion. Every student remember, septum completion indicates that the two chromosomes moved towards the end of the pole and cytoplasmic contents are completely segregated into the two cells and there is a splitting occurred completely. So friends, septum formation indicating two chromosomes are moving opposite to the pole, septum completion indicating that cytoplasmic contents are also equally distributed, ribosomes are also equally distributed, the cell getting split into two. Okay, I want you to remember, okay, the process begins with the cell elongation which require careful enlargement of the cell membrane and the cell wall in addition to an increase in the cell volume. Now friends, you people has to remember the name of the protein compulsory. The cell starts to replicate its DNA, correct, right? Friends, if the cell duplicated its DNA only, one cell can split into the two cell. Now, what is the statement just I have given to you in the previous slide? I told you that, friends, whenever there is a formation of a septum which indicates that chromosomes are moving towards the pole. Now, the question to all of you friends, if this particular septum wants to be formed, there will be a requirement of an important protein. We can call it FITS protein. This is the question already asked. Along with the FITS protein, there is another protein. I will tell you in the next slide. MIRB, M-R-E-B. Please friends, remember these two names without fail. FITS protein and MIRB protein. These two proteins are essential for the formation of a septum. Please don't get confused with that, friends. Okay, now we all used to read one common statement every student remember. That is the generation time of the E. coli is 20 minutes. This is the statement which we use to read. Now friends, the interesting point is out of this 20 minutes, replication will occur very fast, friends. Very fast replication occur. But you know which takes greater time. More than 15 minutes, septum formation only occur. Okay, septum formation only takes more than 15 minutes. But friends, even though septum formation is a slow process, but it is a very accurate process. Without septum formation, chromosomes can't move towards the pole. Now every student, please look into the diagram without having any kind of a confusion. Friends, listen everyone. This is the cell wall. And you can see here friends, this is the cell membrane, and you all can see, right, this is the DNA. And friends, we all know, right, whether it is the prokaryotic organism or the eukaryotic organism, we studied in the molecular biology chapter. All the time friends, the replication is the bidirectional replication. That is what I told to all of you every time. Now every student remember, so this is one fork moving in the clockwise, this is the another fork moving in the anti-clockwise. Now the point is very clear. I have clearly told to all of you, first which is getting elongated, you can see very clearly friends in the diagram. Initially what you can see friends, initially you can see the circular DNA. Now you see here the DNA got elongated friends, sorry, cell, initially the cell is circular. Now the cell wall got elongated, friends, the cell membrane also elongated. Now every student look into the important point. You can see here, right, small green color dotted like proteins. They are only friends FITS protein. Everyone remember my statement. Initially friends, the FITS protein is located uniformly throughout the cytoplasm. Once again listen. Initially all the FITS proteins are distributed uniformly in the cytoplasm. That is what we studied, is it? Now every student remember friends, here what is happening, whenever the cell wall and cell membrane elongate, all the FITS proteins which are present in the cytoplasm, they will go and place in the center. Where exactly the word center means, in this picture you all the students can look into the picture clearly. Friends, please remember, this is called cleavage furrow. We read in the developmental biology. I hope every student remember. What do we call it as? Cleavage furrow. Correct na? Friends, exactly at wherever the cleavage furrow formed, you can see here the FITS protein can form a ring-like structure here. The moment FITS proteins formed a ring-like structure, you can see here friends, the two DNA got duplicated and they completely moved towards the ends. So what I want you to remember for the exam, FITS protein or FITS ring directs the formation of septum. Septum is the one which is going to divide the one cell into two cells. Now friends, here comes the more interesting point. Once there is a formation of a septum, every student remember the DNA got passed to the two daughter cells, cytoplasmic content passed to the two daughter cells, ribosomes passed to the two daughter cell, and friends, the most importantly, even FITS proteins also passed equally to the two daughter cells. Do not get confused with that particular point also. FITS is moving, nucleus is moving, DNA is, not nucleus, DNA is moving, as well as the cytoplasmic content is also moving. Please don't get confused with this particular point. So friends, whatever the shape of the bacteria may be, whether it is the circular or whatever the shape it may be, whether it is the rod shaped bacteria, first friends, there is a formation of the septum occur. Then after the formation of septum, with the help of a FITS protein, the cell can split into two. Very simple point. There is another important protein I want you people to remember here. Every student remember, please don't get confused. One is the FITS protein, another one is the MIRB protein, and the third protein I want you people to compulsory remember, friends, PBP3 protein, which we can call penicillin binding protein 3. Just remember my statements very carefully without having any kind of a confusion. Okay, just remember, friends, this is a rod shaped bacteria, correct na? This is a rod shaped bacteria. Very simple, friends. If this rod shaped bacteria, listen my words, the rod shaped bacteria during the time of a binary fission, if it wants to maintain its width, what do you mean by that? Example, if this rod bacteria has given two daughter cells. Now friends, if the two daughter cell wants to maintain the same width as that of the parent cell, so obviously it requires the protein called as MIRB protein. So MIRB protein, friends, please remember, I want you to remember this particular statement for the part B question. MIRB protein exactly perform the functions of actin. Actin is required for the strength, actin is required for the movement, actin is also required for the maintenance of the size and shape of the cell. Similarly, the MIRB, which is present in the rod shaped bacteria, required for width. Don't confuse and don't forget, friends. And I already told to you that FITS protein is required for the septum formation. Please remember, friends, FITS protein, it is a highly energy dependent protein. Please don't get confused, friends. And the point is here, FITS is not using the energy from the ATP molecule. FITS will always use the energy, friends, from the GTP molecule. Please do not get confused with that. FITS do not hydrolyze the ATP. FITS always use the energy by the hydrolyzing the GTP. There is another important protein, friends, we can call it PBP3 protein or penicillin binding 3 protein. You all people commonly know very well. We read that bacterial cell wall is made up of what, friends? Peptidoglycan, correct na? Now we know very well the peptidoglycan is made up of the NAG and NAM. When this particular NAG and NAM associated with each other by a beta-1,4 linkase, then there is a formation of the peptidoglycan. Now everyone just listen. Okay, peptidoglycan got formed. But friends, everyone please understand the statement. Once the peptidoglycan cell wall is formed, if the cell wall wants to be stronger, it should be added by the sugars. Always remember, friends, peptidoglycan is the major component of the bacterial cell wall. If this peptidoglycan wants to become stronger and if it wants to provide the mechanical strength to the bacteria, it has to added by the sugar. Now, how does it added by the sugar? By the protein called as a PBP3. So it is just involved in the transglycosylation, which means transfer of the sugars. Please do not get confused with this particular point. Okay, so once again quickly remember, MIRB required for width, FITS protein required for the septation, which is required the GTP, PB3 protein is required for the transglycosylation or addition of the sugars to the peptidoglycan to make the bacterial cell wall to become stronger. Okay, so these are all the list of proteins I want you people to remember. compulsory. Okay, so friends, in the tomorrow class, what we do is we will finish the 4 models of the numericals. Okay, I will just finish only 4 models of the numericals. We don't get it, but we will read it. Okay, we will discuss the 4 models. Okay, and friends, please listen to me. Only for tomorrow I want you to attend 2 classes. Okay, only for tomorrow. Okay, one class is at 10 a.m. okay, in the 10 a.m. class we will discuss about the 1 month strategy. Okay, we will discuss 1 month strategy, and in the 3:30 p.m. we will discuss the numerical part, and we will finish the RER as well in the tomorrow class. So by that tomorrow this 4 topics of cell biology will be finished. Then I'll move to immediately to the other chapter. Okay, I hope it's clear for everyone. If you have any doubts, you can ask me. Is it clear, everyone? Please let me know, is it clear? Okay, so please revise it once again without fail. Okay, so in the tomorrow class I will discuss the 1 month strategic preparation. Hope it's clear for everyone. Okay, perfect. Okay friends, that's all for now. I'll meet you in the tomorrow class. Okay, thank you all. I'll see you tomorrow sharp at 10 a.m. Okay, 10 a.m. I'll see you. Thank you all. Thank you.